Very happy to have Paul Jones with us this morning. Paul is Director Population Genomics at Illumina. In that position, he has taken care of really large-scale genomic programs, genomic medicine programs, especially when it comes to marrying, so to say, clinical care and translational research into those large genomic programs. Paul has a long experience in different organizations in the life science industry or life science area. He has also been CEO of Genomics Enterprise and has been head of the commercial operations for the 100,000 Genomes project. And previous to that, he's been working at different companies, and just name a few: Cisco, IMS Health, or Novartis. Paul this morning will talk to us on the functional capabilities that organizations will have to consider and take care of actually when they address programs that address those large-scale genomic programs. Thank you very much, and welcome to stage, Paul.
Thank you very much for the introduction. Thank you very much for the invite. I wanted to just talk today, if I can, around this principle of population genomics and actually just introduce an idea around innovation and how the whole innovation agenda can be spread and where it might start, and how innovation in one area can open up doors to innovations in other areas. And for me, genomics, and particularly the population genomics potential, is as a result of huge innovation over the last 15 years in bringing the cost and the speed of sequencing to something that it is today, where 15 years ago we were talking about billions of dollars to sequence a single genome that may have taken multiple academic institutions across the world multiple years to create an output, to a world today, some 15 years after that first sequence, we can talk about doing multiple genomes in a whole day at a cost of probably not far off $1,000. And it introduces this wonderful concept that I heard articulated in a book by Stephen Johnson called Where Do Great Ideas Come From? And he talked about the principles of innovation and how innovation in one area can open the door to adjacent possibles, as he described it. And I think that's an interesting theme for the keynote today, is just to think about how innovation around the area of core capability in sequencing, around genomics, can open up a door to something like the application of that to the clinical realm at a country level. And I was certainly involved for the last 3 years prior to joining Illumina just a couple of months ago in one of the world's leading endeavors around population genomics, population sequencing, in the 100,000 Genomes project in the UK. And I'd like to bring some of the themes out from that innovation around genomics, how it opened the door to the potential for population genomics, and indeed some of the key lessons that were learned from my 3 years there that might be applicable to equivalent endeavors elsewhere around the world that could be relevant to all of us as we're users of the healthcare system in the future. So that's just kind of, if you like, an opening thought provoker. Please bear in mind this principle of open innovation, adjacent possibles, and the idea that innovation in one space can open up a plethora of opportunities elsewhere. The topic, as I describe it today, is this principle of looking at how we scale these initiatives. And I'm actually just going to use a definition around what we might mean by population genomics or population or genomic medicine. And it's this principle of bringing translational research, which is often couched in an academic or research agenda, and applying that in a clinical setting in order that it can have an impact around diagnosis or the potential for new treatments and actually have a patient benefit. So it's this critical link between the research academic agenda and the clinical domain. The use of this in everyday practice. And that was one of the underlying principles behind how the whole 100,000 Genomes project was initially set up. So really one feeds off the other. And that's really the key element here about looking at adjacent possibles as a theme. And I'll summarize it here in terms of this principle of something that may be initiated in clinical research that then will translate into clinical care, but at the same time, the feedback that comes from the application in a clinical setting then informs the research agenda. And I think there's a beautiful potential for those two to work very effectively together and actually to accelerate the speed with which things move around those two circles. And that's one of the things that we were striving to do in a UK setting with the 100,000 Genomes Project is I think there's a rough rule of thumb that says to get any great innovation that may start in an academic or research setting into clinical practice can take upwards of 20 years as a rough rule of thumb. And the key challenge is how do we accelerate that? And actually accelerate it not from 20 years down to 15, but actually take it down to something like a 2 or 3 year time frame and make sure that we continue to close those loops so that one constantly feeds the other. That was the challenge that we took on in the 100,000 Genomes Project. And indeed, I'd love you to think about the equivalent challenge in your own market, in your own domain, where that acceleration and that link and that translation synergy can actually change the rules of the game. So the 100,000 Genomes Project, for me, was a great example of genomic medicine at scale. The topic of this keynote today is about what lessons came from investing in that agenda that means that we can start to utilize the lessons elsewhere. And one of the key things that I would highlight is that they started with a range of very, very clear objectives. So it was really looking at this principle of the combination of a large heterogeneous population with a great academic research base, but also with a clinical system that was functioning from a national perspective. The NHS, as we call it in the UK, had the potential to make an impact to prepare for a future of genomic medicine. And we were looking really at how do we use that program to drive insights and understandings around new treatments, new diagnostics and new devices. So how do you use that system to accelerate the development of those key principles that would benefit patients? We're also looking at introducing this into the healthcare system. In general, the healthcare system was not prepared for a genomic medicine future, other than from within a few specialists that were experts in this space. We wanted to broaden this whole agenda so that the whole of the healthcare system was prepared to accept and indeed accelerate the use of this endeavor. And the third core agenda, which really spoke to the role I had as the CEO of what we call Genomics Enterprises, which was running the commercial interface, was to explore how we drive and encourage private sector investment and commercial activity around the whole genomics agenda. And I'll come on in a second in terms of some of the specifics around that agenda that we were able to deliver over the 3-year period. The key point I would emphasize is that all of this was enabled through citizen or public support. Without being able to demonstrate that we could take due care and attention of patients' stroke citizens' data and manage that in a way that demonstrated the impact back to those individuals, but also were very open about the other stakeholders that we had at the table that would contribute towards making this useful to them. If we lost that support, the whole program would fall. And so we were extremely conscious about managing that data aspect and the protection of the privacy of the individual in a way that made it very, very clear what our agenda was. And again, that will be a theme that I return to over the next half an hour. By the way, if there are any questions as I go through this presentation, please feel free to stop me at any point. I don't need to wait for the end to Q&A, but at the same time, we will have a specific Q&A at the end of the session. So let me just, for those, how many people in the audience here are very familiar with the Genomics England agenda? It's something that everyone has heard of. A few nods around the place. The terminology around population genomics is one that doesn't necessarily translate well into other cultures, other countries, but the principles of it are really about this link between the academic research endeavor and the clinical applicability. There's a data core to it, and actually I'll talk in a second about some of the core principles that would emphasize to me, anyway, what makes a population-based agenda. But just to give you, for those of you who weren't aware or aren't aware of the 100,000 Genomes Project, this was initiated in the UK after the Olympics in 2012, sponsored by the UK Prime Minister, who encouraged the contribution of some funding to look at how the UK could build on the success of the Olympics and use another endeavour to drive the positioning of the UK as a place to do research, but also, and perhaps more importantly for David Cameron, the principle of then being able to apply that to make a difference to patients' lives. There are a few key themes around how the endeavour was set up that I think provide some interesting insights into how these kind of initiatives could be explored in other markets. And the first of which, for me, is absolutely central to this, is this principle that we were set up as a company. So Genomics England Limited is a separate and distinct organisation from the healthcare system in the UK. Now, the shares of that company are owned by the Secretary of State for Health, but we did have a separate and a different agenda. We were set up as a commercial organisation, even though it was wholly owned by the Department of Health. That's a key theme in terms of enabling the driving of innovation in light of the challenge of operating within a live and functioning healthcare system. There's some interesting lessons that come from that I'll revisit time and time again over the next few minutes. We had a very, very clear agenda. Right from the outset, it was very clear that we would aim to sequence 100,000 genomes, whole genomes, within a 3-year time period. Now, we were talking primarily around a period that started in 2015, 16, and 17. So we were looking at a 3-year period to sequence 100,000 genomes originally. Now, over time, and I will come on to this, there were 1 or 2 challenges in 1 or 2 areas, and the timelines have slipped slightly, but that endeavour, the objective still holds true. The objective of the Genomics England programme is to sequence 100,000 genomes from a population in the UK within a slightly more flexible time frame than was initially anticipated. That is still absolutely on the cards. And again, a key lesson for me going forward is having that very clear objective makes a huge difference in terms of highlighting progress and making sure particularly the patient citizens are on board with what you're doing and how you're progressing. It was, by definition, a clinical service. We were at the interface into the healthcare system itself. That's where the patients were consented. But at the same time, there was a huge research endeavour. So it was this classic link between the research and the clinical that was central to the success of the programme. Very early on, we were very explicit about where we would apply our initial focus. And we chose rare disease and cancer as 2 areas that seemed to be areas where we could make a difference to patients' lives within a relatively short time frame. That wasn't to say that we didn't have numerous requests to apply the principles of the programme to other areas too, but a very clear focus was determined right at the outset, and we stuck to that around saying, we know there's the potential for applicability elsewhere, but we're actually going to stay with rare disease and cancer to start with. Once the 100,000 are done, there's clearly, assuming it's successful, the potential to apply it elsewhere, but for the purposes of the initial programme, focus on cancer and rare disease. It is absolutely explicitly based on consent. So when patients are recruited from the healthcare system, it is very clear to them what they are consenting to. And we actually went through quite a rigorous, innovative process in itself of securing a national consent in the UK that meant that when individuals were recruited from the numerous centres across the UK that they were all signing up to a single national consent. And that made a huge difference towards the principle of being able to accelerate the various stakeholders who were looking to engage in this programme without having to go to each local centre to renew the ethical IRB approval process. So that was one centralized process. Again, a very interesting lesson in if you're looking to do this at scale, that principle can really help accelerate these type of endeavors. The core agenda was the 100,000 genomes project, but actually when I look back, I think the more challenging aspect of this was around the collection of the clinical data, the phenotype data. Both of them were absolutely critical. We could not do one without the other. And as I look back, and again, reflecting on some of our bigger challenges around the table, the genomic sequencing piece was probably something that was less problematic than the difficulty and the challenge of collecting both existing data from a healthcare system, but also creating data models and collecting new data specifically for the 100,000 Genomes Project, particularly around the rare disease agenda, that previously hadn't existed. So we had to create new data models. We had to ask physicians who were recruiting patients and consenting them into the program to also complete and advise us initially and then complete data models that would help us collect and capture the right information around the different disease areas that we're focused on. And if I want a range of the stakeholders that engaged with the program, particularly from the industry side, said to me on more than one occasion, Paul, if you do nothing else, whatever happens with this program, just the very creation of rare disease data models that previously didn't exist, and the capturing of critical phenotype information from the healthcare system itself is a huge win, a huge plus, and something we're prepared to invest to enhance and continue, irrespective of the value that might come out of this program in the future. So it's interesting, there are things that could become quick wins in a program like this without even realizing that that was one of the core objectives. We actually set up and formalized through a tender process that went national the identification of 13 different, think of them as being spokes on a wheel, that were really the recruitment centers that were allowed us to pull into the hub at Genomics England where we would do the sequencing. And so there were actually 13 centers set up that covered the breadth of the UK, and there tended to be one major center in each of the major regions. And that center was as a critical focus around which we both recruited patients in the first place, but also to which the feedback would go at the end of the program. Well, actually not the end of the program, once an interpretation endeavor had happened and we were able to confirm a diagnosis and make recommendations around treatment. So they were the start and the end of the process. And I will in a second just highlight exactly how the process worked. Those 13 genomic medical centers in some cases would be a single large center. In other cases, there would be multiple centers associated with it. But the key point was we covered the breadth of the country through this endeavor. Now, the data, as I mentioned, was held in a secure and monitored environment, strictly regulated access, but we were very, very explicit that we would have industry in the form of biotech and pharma companies engaging in this endeavor. We named the industry participants the Gene Consortium. They came together as a group of 12, 13, 14 companies by the time we finished, an additional one or two. So companies who were prepared to collaborate together to work with us as Genomics England to articulate what they would consider to be valuable and help steer the program in order that they ultimately could develop new precision medicines and diagnostics in the future. So they were there almost like an advisory group, but actually they were also someone who was accessing the data very explicitly. The patients who signed up and consented realized that that was the case. And the thing that surprised me, having spent 30 years in and around the life sciences industry, is there was a real lack of awareness of the role that the industry can play in furthering the endeavors around precision medicine. There was not a generally highly accepted view that said without the industry at the table, you are unlikely to get new precision diagnostics and therapeutics. That commercial interface was key, and so we were very explicit in reinforcing that message to the patients and the citizens as part of the overall education associated with this agenda. So that was the data asset and the industry interface, there was also a large medical education program that was run with part of the funding going towards educating the clinicians in the system, actually supported by Health Education in England, that continues to this day in terms of a master's degree, supporting education around furthering genomic medicine, and funding that was supporting clinicians in practice and professions allied to medicine to be skilled up in this space. At the same time, we also built an interface through the Chief Science Officer, Mark Caulfield, who ran an interface into what we call GSIP. So the Genomics England Clinical Interpretation Partnerships. And that was, in effect, the academic interface, where what we said is that we've got a data asset that is being built up here at the interface into the healthcare system. And would you, as researchers, be interested in accessing that to facilitate with the interpretation of that data? To look at this link between the genomic and the phenotype data and to help us come up with those answers. We actually had over 2,250 academics from around the world put up their hands and say, yes, absolutely, give us access to that data and we will facilitate that interpretation agenda for you. And so just on that interpretation piece, it's worth reinforcing, because really in a way, and I talk about this principle of adjacent possibles, maybe 15 years ago we would have talked about genomic sequencing as being the big challenge. That was the multi-billion-dollar problem that took forever with huge collaborative efforts required. Actually, in this day, for this program, that was the piece that was probably the smoothest and the easiest aspect of this to manage. It was the output that was created from that in terms of the big data challenge, that looking at the patterns that linked the phenotype and the genotype together, where we really needed support and where the challenge was moving on to. And we almost had like 3 tiers of interpretation. So you had a Genomics England bioinformatics team built internally within Genomics England, a team of 25 people, highly skilled postdocs, many of them, who were focused around making sure that we set this up as a program internally within Genomics England in the most appropriate way. There was a second tier of interpretation around commercial organizations that we could align to engaging with our data set and that would facilitate with our interpretation. And we actually ran a competition to identify those commercial partners that would be most appropriate to facilitate that endeavor for us. And then there was a third level, which is this GSIP level, these 2,000 plus researchers who, again, being given access to the data, they were able to facilitate the interpretation, probably around the more challenging aspects of this, that the commercial organizations were packaging up and already making available as commercial offerings. This was something that fitted more into being published and would be part of a research agenda. So there were multiple tiers to facilitate the challenges around using the output from the sequencing agenda that was really the next phase of activity. And then the final point I'd make is around really the funding commitment. Initially, coming from the very highest levels within government and with the support of Sally Davis, who was the Chief Medical Officer of the UK, actually in her responsibility in charge of the National Institute of Health Research, she initially made £200 million available to support this endeavour that was then supplemented in an additional budget round some 18 months into the programme. So a significant investment from the government and allied institutions, as well as Medical Research Council and Wellcome Trust, to kickstart this program, focused on a single objective around 100,000 genomes, focused around cancer and rare disease, very large, hugely complex program at the interface into the healthcare system, but also including stakeholders from industry, stakeholders from academia, continued political sponsorship, and the setting up of a commercial organization in the form of Genomics England Limited to really coordinate this activity. It's quite a significant investment on all fronts to kind of transform the agenda and move this whole endeavor forward. This is a very brief summary, and this is now some 3.5 years later, that I think is a very nice simplistic view, because there's a huge lot more complexity behind this. But the principle here really is about, it starts with the patient at the top. It's that patient going into a genomic medical center, one of those 13 centers or those centers are allied to those main 13 hubs, that are then recruited into the system, where their sample is taken, where their clinical data is captured, and where permission is given to access any data that exists in the system about those patients. That sample and that data is then managed appropriately. Again, there's a huge amount of complexity behind this in terms of where that travels. I'll come back into some of the detail on that in a second. It then passes over to a sequencing partner. Now, Illumina, I wasn't with Illumina at the time, but I now work for Illumina. Illumina were a major commercial partner for Genomics England, where we, as a company, won a contract to actually set up a lab on behalf of Genomics England to run whole genome sequencing for this program. And that lab today is probably the largest whole genome sequencing lab anywhere in the world. So that was the sequencing piece. Then it comes on to the interpretation agenda that I was defining, where there was an internal, there was a commercial, and an academic interface, with the idea that you'd create a diagnostic report that would go to a clinician, that would then get fed back to the patient. You see the blue and the red box at the bottom here is really this link from the interpretation agenda, where a data archive is created, the research agenda continues there, and from which a knowledge store is created that's constantly fed back into, that contributes into future diagnostic reports. So this is a system that never stops working. This is at the interface into a healthcare system that will constantly be replenished. And just going back to the second core objectives of the program, this is helping a healthcare system prepare for a future where genomic medicine will be at its core, with potentially disruptive implications. Now I've taken this diagram from a report from Sally Davis, Dame Sally Davis, the Chief Medical Officer in the UK, just published early this year, summarizing, as she does once a year, a major theme of interest to her, where she put for this year, genomics on the agenda. And this is a diagram that I've taken from that I think very nicely summarizes a hugely complex program, but in a way captures the essence of the important aspects of this endeavor. So it begs a question then, what makes something genomic medicine? What makes it something that you describe as a population genomics agenda, or something akin to that would be relevant for your own market? And I've drawn out some themes here that strike me just based on my experience over the last 3 or 4 years working in this space. So the key principle is that it has to be embedded in the healthcare system with ongoing patient engagement. That's core to this agenda. The program links research with clinical care, and I hope that point's coming through loud and clear, with the appropriate consent, the opportunity to re-engage the patients. I didn't realize when I started how important that is. This process never stops. The potential through the consent that those patients can be recontacted is really, really critical, because it provides an opportunity to collect more data from them, potentially to collect more samples from them, and also from an industry perspective, the potential to recruit those patients for trials. And so that is incredibly valuable, and was seen by the industry guys who engaged with us at this table as being one of the critical things that really made a difference with this program. It's something I would highly encourage you to do if you have an opportunity to influence the agenda in your respective markets, as being a really valuable thing that benefits patients but also benefits the other stakeholders at the table. The second key theme is really this principle about building an integrated data platform that grows in value over time. It's not just about genomic data, it's about the phenotype data, the medical record data. Ideally, it's about outcomes data that's captured from a prospective perspective, made available not just for a clinical setting, but also for academia too. And then the third key agenda is really this principle of active translational involvement with industry and academia at that same table. I pushed some of the stakeholders that we had around the table to define for me what they considered to be most important. You know, is it about patient consent? Is it about the potential to recontact? Is it about academic involvement? And actually, they wouldn't be pushed, because the principal answer was, it's having everyone at the same table. It makes it incredibly challenging to manage. But if there was one key point I'd like you to take away from today, it's this having at the same table the researchers, stroke academics, the industry guys, political sponsorship, and clinical engagement that makes this what it is. It makes it harder to manage, but having all of those, none of whom that dominate, they all have a voice at that table, makes this a program that is valuable. Just to give you a perspective from where the industry guys were coming from, so this is pharma and biotech, where I went out to the largest 25 companies in the world and I said to them, what would you know what we're doing with this 100,000 genomes project? It was a presentation not dissimilar to the first couple of slides I've given today. What do you need as an industry in order that you'll see this as valuable and indeed valuable enough to pay for? We were looking at building a commercial interface into this. And these are some of the principles that they highlighted as being important. And you can see many of the same themes coming out. They were interested in linked data. They were interested in who we had as bioinformatics experts within Genomics England, but also the other commercial partners that we had at the table that were able to access the data in return to facilitating the interpretation with us. They also were interested in the academics that we were working with, those 2,250 plus researchers around the world, who, by the way, were formed into virtual groups around specific research themes. So there are upwards today of 50 different research groups into which those 2,250 researchers fit. We're trying to encourage everyone with a common interest to work together and for there not to be duplication across areas so that we can keep the research agenda with some degree of focus around certain areas. They were very interested in this streamlined ethics approval agenda. Joint working with the healthcare system. There was a big regulatory question that seemed to fit very well into what does the regulation need to look like in a country to prepare for a future of precision medicine. And then indeed they were interested in saying, if we engage, and initially we may engage as a consortium, they all came together, sat around the same table, everyone shared the same insights, and we encouraged an interface into the academic agenda, into Genomics England. They were providing advice as to what they would consider valuable and putting some pressure in the system to ensure that was delivered. But they were also interested in what future engagement models for the industry might look like. And just to summarize for you in terms of where we ended up, we talked about the consortium being a great basis for the first 2 or 3 years to get the program running, everyone to learn to walk before they run. But if in the future, individual companies then were comfortable with how they engage with something like a Genomics England program, every opportunity in the future to then look at what we call a direct engagement model, a specific company with a particular interest in extreme phenotype, maybe with a drug in development that they'd like to recruit patients to trial for, coming to Genomics England to say, could I please negotiate a specific engagement with you as a company in private, probably not sharing the insights until we're ready with the rest of the consortium and the other stakeholders, and we would absolutely be open to a direct engagement on that. Again, with the principle that the result of that would potentially be patient benefit.